My name is Laura Andre, and I'm the now current chair of the ALBA network, following in Megan's footsteps. And it is now my great honor and privilege to introduce the winner of the 2022 ALBA Elsevier Award. This award goes to Dr. Elena Avale, who is an assistant professor at the University of Buenos Aires in Argentina, and also a group leader at the Institute for Research in Genetic Engineering and Molecular Biology, which is a very long title, also known as CONICET in Argentina. Elena did her PhD in Buenos Aires and then went on to do postdoctoral research in Europe. So she spent time as a postdoc at the Institut Pasteur here in Paris, in Jean-Pierre Changeux's lab, and then actually moved on to the UK, to my very own King's College London, where she did a postdoc with Jean-Marc Gallo. She's kind of moved around from looking at dopamine receptors through to nicotine addiction in her postdoc in Jean-Pierre Changeux's lab, and then at King's, moving into the field that she's really been pursuing ever since in her own lab, which is looking at tauopathies and the role of tau in neurodegenerative disorders such as Alzheimer's and Parkinson's. She's really made some incredible contributions in this field, highlighting, tau is a now, once again, kind of, hot protein in the neurodegenerative disorders. Not my field, but I'm sort of vaguely aware of this. But she's got some really beautiful work looking at how tau works and some, really innovative approaches to thinking about how we may be able to modulate different isoforms for the treatment of these disorders. She's done all of this based in Argentina in a landscape that is really quite challenging in terms of funding and support and networks. It's really a particular pleasure to welcome her to the stage now to tell you about her fantastic work. Thank you.
Hola, hello. So thank you very much to ALBA for this recognition. I think it's, as Laura says, being in Argentina, as you can imagine, it's a big challenge, but it pays off when you see the students, like my student that's sitting there, here in an international conference. So if I have to make a balance of how life is going in Argentina, I will say, you can imagine it's hard, but it's worth being there and trying to at least do a little bit for our research. So I will take the opportunity of being here. You know dementia, I don't have to introduce that to you here, but just to highlight that it's a huge problem all over the world, and Latin America is not out of that. It used to be said that dementia was a problem of rich countries or developed countries, but it's not that today. We know dementia is spreading all over the world, and if you see, low and middle income countries are now having increases in dementia. So this is something that's very important for researchers and for governments to pay attention to. And in Latin America, there is an interest to go towards research in dementia, of course, but we have this little problem that we don't have enough funding. And I know you know this, but the numbers are really frightening, because this is the amount of money that has been invested in science in Argentina between 2008 and 2018. So just to figure out, as I will tell you later, when I went back to Argentina, we were here, so everything was like, wow, yeah, we can do it. But, well, things are not that good now, but still, we did it. And after a lot of massive protests during the last years, last year we got the first law approved by the government to increase our GDP investment in science. So we are now, last year we were at 0.28, and we were promised to achieve 1% of our GDP by 2032. So before retirement, I hope I will see that. And fingers crossed they will do that. Of course, it won't be at the levels of investment in other countries, but hopefully we will be able to increase science. And having said that, I have to say that we do have good neuroscience in Argentina. There are many courageous people doing very good neuroscience there. I don't know why I am here, because there are many others that are much better than me. But well, I was lucky to be here, but I want you to think about that Argentina is doing important contributions to neuroscience. So when you think about organizing conferences, we were discussing with Laura today, think about Argentinian neuroscientists, because nice things are going on there. So having said that, let's move to science. And yes, as Laura said, I moved around many fields, always in the brain. But the brain is gorgeous, so we can do a lot of things, and we are always fascinated about it. Until during my last postdoc, I met tau, and I fell in love with tau, as many others, and I decided to continue my career on that. So what I'm going to tell you today is what I have been doing since I did my last years of postdoc at King's, and how I continued that in Argentina. So you all know that tau is involved in dementia, and not only in Alzheimer's disease, but many other dementias, which are named tauopathies. We are going to go on that soon. But the first description of tau, it was not that tau at that moment, was done by Alois Alzheimer when he described this accumulation of insoluble protein inside neurons. And these are what today we know as tangles, neurofibrillary tangles. So many work has been done in the tau field. Tau was described as a microtubule-associated protein in 1975. And then 80 years later, after Alois Alzheimer first described tau, it was discovered that it was the bad girl in Alzheimer's. It was an accumulation of tau. The neurofibrillary tangles that Alzheimer has described were actually accumulation of insoluble tau. So now we know that tau is involved in many diseases. They are all named tauopathies because they are related to abnormal accumulation of tau, and is one of the most frequent causes of dementia. We have dementia without tau accumulation, that's true, but the most frequent cause of dementia is accumulation of abnormal tau. And this is what you can find today as an electron microscopy for a neurofibrillary tangle. And when I see this, I say, okay, Alzheimer was really clever because it's quite similar, no? It's amazing. Okay, so if we have to classify dementias, we can, of course, put first Alzheimer's disease, which is the most frequent, but then we have no AD tauopathies, which do not accumulate A-beta extracellularly, but all these diseases accumulate tau inside the cell. And the clinical symptoms are related to where tau is accumulated, so it can be more cognitive behavioral or more motor diseases, as we are going to see in a minute. So the tau dogma, and we know a lot about that, is that tau is a microtubule stabilization protein. So its main role is to do that, to keep microtubules healthy and tight, so the cytoskeleton is okay and everything is going fine in that neuron. And when tau detaches from microtubules because it is phosphorylated, mutated, or whatever happens to it, it will accumulate in NFTs, but at the same time, microtubules will collapse. But a lot of work has been done in the last decades showing that tau is not only a microtubule stabilization protein, and it has essential roles in neuronal physiology. I'm not going to go into that now because I don't have time, but it's fascinating because every week we have an article showing a new function for tau. So I used to say that it's a multitasking mother, because I feel like she's doing all the time the same things all the time. So one of the of the most interesting functions of tau that has been described is its role in axonal transport. Not only to regulate the movement of kinesin and dynein, but also to detach motors that are moving in the axon. So, just to summarize all the functions of tau, if you can imagine inside a cell, tau could be bound to microtubules, could be in spines, could be doing many things at the same time. Just to keep that in mind, it's not only microtubule stabilization. So, my work focused more in what's going on when tau undergoes abnormal post-transcriptional regulation. So I will focus on this part. This is the MAPT gene, which is transcribed to the primary RNA, and then we can have alternative splicing of exons 2, 3, or 10. I will focus on this part because it's what we have been investigating, and particularly exon 10 can be either included or not in the mature transcript. So in that case, in a normal human neuron in adulthood, we have equal amounts of 3R or 4R isoforms. And these 3 means that we have 3 microtubule binding domains, which are encoded by these exons 9, 11, and 12. And if exon 10 is included, we have 4 microtubule binding domains. So you can imagine that with 4 microtubule binding domains, attachment is stronger than with 3 microtubule binding domains. Okay, so in addition to that, after translation, tau can be modified, of course, by many mechanisms, and phosphorylation is the most well understood. Not that well, but we know a lot about tau phosphorylation for physiological function, but also, and here I put in black, because it could be an abnormal hyperphosphorylation of tau due to many reasons that will lead to tau accumulation, right? Okay, so my work focuses on RNA, so I'm not going to enter in the protein processing because it would be impossible. And what I'm going to summarize today is the main achievements we had when trying to modify tau isoforms by a strategy called RNA transplicing or mRNA silencing of tau. So let's start with this first part. Just to refresh, alternative splicing of the MAPT primary transcript is regulated by cis-actin elements, which are in exon 10 and in the first part of intron 10. So this is the normal loop that should be formed after transcription. And many proteins, trans-actin proteins, will bind to these regulatory regions to decide if exon 10 should be included or not. So, and you know, the transcript is the same in every part of the brain at every moment of life, but this regulation of alternative splicing depends on the presence of different trans-actin factors. So, in humans, in humans and in rodents, during development, we always have only 3R tau, so exon 10 is always excluded. When we reach adulthood in humans and close primates and non-human primates, we have equal amounts of 3R and 4R tau, while in mice and other mammals, we only have 4R tau. And this, as I said in the past slide, this is decided by the transcription factor, transplicing factors which are there. So, this is what we expect in a normal brain of humans or mice. But in humans, things can be wrong, as it always happens. So we can have many mutations in the MAPT gene, either in exons or in particularly in this intron that I mentioned that it was very important for splicing. And all these mutations lead to disease. It means abnormal tau accumulation. And this is tricky because if we have mutations in an intron, in principle, we do not expect to have a problem in the protein, but we do have because when these mutations affect splicing, we can have an imbalance between 3R and 4R in the brain. So the two isoforms are normal. They do not carry mutations, but the balance between them is altered. And this is what leads to disease in many tauopathies. So, given this problem that there could be an abnormal exon 10 splicing due to mutations, and not only mutations in MAPT, but also it was described that abnormal splicing could be due to environmental factors or mutations in splicing factors. So there are many reasons that could lead to an abnormal tau isoforms content. When I was doing my postdoc in King's with Teresa Rodriguez helping me a lot, I'm always grateful to her, I learned how to do this transplicing strategy, and the challenge was try to use it to change the pattern of isoforms splicing. Tau isoform splicing. So, in a normal situation, the cis splicing will lead to either 3R or 4R, everything in gray here, just the endogenous splicing. But when we interfere with that by expressing this molecule, this is an artificial RNA molecule that can bind to the endogenous RNA, and this will be taken by the endogenous machinery of the cell to do a transplicing. So if this happens, the endogenous transcript will finish here, and after transplicing, we will have this artificial RNA included in the mature transcript. So this will lead to this chimeric RNA. And this could be done either to force exon 10 inclusion or avoid exon 10 inclusion. So the big challenge, as I said, was to make it work in neurons. So I started doing with the primary mouse neurons in culture. After many trials, finally we got a construct that was able to attach to the endogenous primary transcript and lead to transplicing. And the splice junction was okay, and this could be translated to protein, and everything was very happy, and we went to the pub and we got a beer. But then the big question was, okay, we can do it in primary neurons and culture. We want to do how it works in human neurons, which are the consequences, and if tau pathology could be precluded with this system. So for personal reasons or many other reasons, at that time, when I just finished that work, I decided to move back to Argentina. Many people was saying me that was suicide, but I'm very tête dure, comme dit le français. So I moved to Argentina, and thanks to the help of many colleagues there and very motivated students, we moved forward to this project. So first thing we wanted to answer was that, okay, can this system be done in human neurons, and what's going to happen if we modulate tau splicing in human neurons? With the help of my colleague and friend Tomás, in human neurons in culture, we did the transductions with these PTM constructs, and then we checked after being sure that we had changed isoform balance in neurons. You know, they are human neurons, mature human neurons, so we expect this is the normal situation, and this is what we forced with the system. And we did first morphology studies. Everything was fine. Even the change in isoforms was there. It was done when neurons were already mature. But when they check, because they are the experts in this field, the outcome of axonal transport was really amazing, because in the normal situation, molecules move around happily, and when isoforms imbalance is there, it's really impaired, the axonal transport. These are not very fancy movies. They have been done many years ago, but I think it's quite robust, the results, so I always like to show it, because it was the first readout that endogenous isoform modulation could change a physiological function in neurons. And okay, well, then we characterize how was this change specifically done. I'm not going to enter on that. So, well, we know it works in human neurons. We know it can change a physiological function. Now, the big thing was to try to make it work in a rodent brain. So we took advantage of this mouse model, which is the human H-tau mouse. It does not express murine tau, but instead it has a full-length human construct. They have cognitive deficits between 9 and 12 months. It has been well characterized before. And what it was hypothesized at that time when we started the project is that there is no mutations in this transgene, so the phenotype of these mice should be or could be due to this isoforms imbalance because the combination of the human transgene with the splice factors in the mouse brain led to 3R excess compared to the normal 4R tau that is expected in mice. So we analyzed the brain of these mice. It had been done before, but we tried to reproduce in our lab, and we have seen that the prefrontal cortex was the area more affected by tau accumulation. So my first student project was to, in these mice, try to prevent phenotypes by modulating tau isoforms with the PTMs, the molecules that I explained before. So, well, this is just to show you that we did it in the brain. It was possible to modulate tau isoforms content in either at the RNA or protein level. And we observed that mice that have been treated with these molecules to balance tau had lower accumulation of hyperphosphorylated tau, either by immunohistochemistry or Western blots. Also, electrophysiological patterns were recovered. This was done in collaboration with a colleague in Argentina. And we could prevent the cognitive decline that had been described in these mice. So this was very interesting for us because even if it was a prevention, it was possible to avoid or to preclude the cognitive deficit, which is the most important readout of dementia in the model. So this is what we published in 2018, but we at that time became also interested in other phenotypes of these mice. And this was due because during the characterization of the model, the students were trying to do the Morris-Water maze, which had been described as a phenotype in these mice, but they said they were very bad swimmers, so something was going wrong there. So we decided to go to motor coordination evaluation in the Rotarod, and exactly, these mice were terrible in the Rotarod. They were falling all the time, even worse than the tau knockouts. Tau knockouts had been before described as being bad in the Rotarod, but these were even worse. So we tried to check if something went wrong with dopamine contents, because as Laura said, I had been working in dopamine during my PhD, but dopamine contents were all right in these mice. So also we could not recover the phenotype with L-DOPA administration. So we were thinking that this could be a possible model for atypical Parkinsonism, which is a phenotype related to tau pathology in many diseases such as PSP, progressive supranuclear palsy. So we scanned the brains of these mice, and exactly the striatum was really affected in metabolism analysis. Also, electrophysiological recordings in the striatum of these mice showed a deficit, but there were no phospho-tau deposits, and it was challenging because there was no tau accumulation, but still we hypothesized that the tau imbalance would be detrimental even in the absence of deposits. So my second PhD student, who is Anna, many of you know her, she did exactly the same that we had done before for the cortex, but now in the striatum, and Caro, who is sitting here, helped her to do all the molecular analysis, and we could recover isoforms balance in the striatum, and we have seen that the Rotarod behavior was recovered to wild-type levels. Here we see, and this is the control H-tau mice. And also, electrophysiology was already partially recovered after the injection of the PTMs. So with all these, we said, okay, again, H-tau phenotypes in another structure could be precluded by early modulation of tau isoforms. But the big question was, okay, prevention is okay, but we need to cure disease. We are not trying to cure disease, but we are trying to do something towards that way. So last year, Caro and Javier decided to take this question and say, okay, we are trying to do this, but later on, when phenotypes are already there, so we discovered that at 6 months, we already have some behavioral phenotypes, either related to cortical or striatal deficits, novel object recognition, or rotor role. And also images showed that between 6 and 9 months, already there was a change in the prefrontal cortex, but in the striatum was a bit later. So 6 months was a good moment to do an intervention. And that's what we did. We injected mice either in the cortex or in the striatum with these constructs. And we have seen that phenotypes were recovered. In the red here, we see this is the control injection, and this is the recovery in the novel object recognition, and this is the recovery compared to the control in the number of folds in the rotor. So the answer that could phenotypes be rescued after phenotypic onset was yes, and that was very good. We are happy with that. And well, with all that, we are now trying to publish our article, and hopefully we'll be there soon, fingers crossed. So just to finish, because I'm late as usual, I will just give a little bit bite about what is going on now in the lab. In addition to modulation of tau isoforms, we are trying to tackle this idea of tau silencing as a prevention or rescue of tau pathology. So as I said, tau is important for physiological function of the neuron, but when tau is wrongly processed, it leads to disease. So it's a balance between, okay, we don't want to take all tau out because we need some tau in the neurons, but if there is an accumulation, this neuron will die. So we need to play with that balance. So we developed a new tool, a microRNA, to silence tau, and it works nicely in vitro. And then Caro injected them in the prefrontal cortex. This is last 6 months, I think. You see her. As soon as she could go back to the lab, she started to do these experiments. And this time we decided to inject either early, before onset, it means at 3 months, or late at 6 months. And what we have so far is that with early injection or late injection, in both cases, we can have a recovery in the novel object recognition compared to the control H-tau mice. So you know the novel object, this is the random choice of novelty. So it seems that these mice could recover this preference for the novel object. And also we observed that phosphorylated tau is decreased when mice are treated with the microRNAs. And this is, I'm serious, this is from last week. We got the first scans of these mice, and we see that in the H-tau, there is a huge change in glucose metabolism, and it seems that the prefrontal cortex is clean in treated mice. But this is work in progress, so we need to check this. But what we think now is that local tau silencing could improve behavioral and biochemical phenotypes. However, we still need to check if this works in human neurons and which are the functional consequences. So we still need to investigate more the pros and cons of tau downregulation. So maybe in a couple of years, I can tell you about that. So this is my group. I already presented them during the talk. Sonia and Anna already finished their PhDs, and this is my current group. And thank you very much to all the people that gave us this support, which was very important to do our research, and the collaborators, most of them in Argentina, who are always open to help us to do this multifunctional analysis of our mice. Thank you. I still have 20 seconds.
Thanks very much for a really amazing talk and very entertaining as well as striking to see, how little money you managed to achieve all of this with the funding situation in Argentina. So we're almost out of time. We've maybe got just time for a quick question, if anyone has one. Wave at me.
I will be around anyway, so if you want to discuss.
Well, I have one very quick question, which is, I wonder, do you have any understanding of how it is that this imbalance between 3R and 4R actually leads to these the pathology?
Stoichiometrically, you mean?
Yeah, because there seems to be, obviously you see it, you can adjust the balance either way. There's a kind of bidirectionality going on. And indeed in the mouse, you would, if it was just the straight numbers, you would think that it would have an abnormality, but actually when you shift it, then it changes.
Yes.
So I wondered just what you think.
Yeah,
of the pathology.
actually I have to be honest, we wanted to do it all towards 4R tau, but we couldn't. Even when increasing the amount of viral vectors we injected, it was impossible to get all 4R isoforms. But still, our hypothesis is that as we are with the human MAPT gene, a balance, it's okay. So for the mouse, it can cope with it and have a normal function of the brain. But ideally would be to do all 4R, but honestly, we tried and we couldn't.